Okay.
Okay.
Okay, so I'm trying to share the screen. Okay, I see that. And can you see my screen?
Yes.
Super good. So the floor is yours, Oleksandr.
Okay, thank you. Second attempt. Well, what I have to say, like, I'm feeling, just to get rid of a feeling of being imposter on this meeting, I have to warn you that I am not a specialist in animal health, in human health, and I'm rather a zoologist or ecologist representing here academic science. I'm a researcher at the university and I'm teaching students. And somehow I would like to shift the focus in One Health, in discussion of One Health, from the human and agriculture animals to wild animals. What we will see if we will look on the issue of One Health from this side. And of course, I'm not trained and I'm new in this field, comparatively new, I believe, in this field. And what I did first, I did the research and tried to figure out what is One Health. And what I found first, please, next slide, that there is a huge discrepancy, of course, in how you define it. One is approach to design and implementing programs, policies, legislations, etc., official WHO definition is too broad and unspecific for me. And next one, please. Next slide, please. Is good, but what is optimal health for people, animals, and our environment? Optimal health may be viewed differently by different specialists from different fields, and diseases which are considered to be bad for human, for agriculture, actually are also playing some crucial role in population and number regulation in wild populations. I think it's obvious that for example, in the situation with African swine fever, we have this disease not only because it was brought from other continent, but because the number of wild boars in environment in Ukraine is way too high and is not regulated by natural predation, which is missing because of a human hunting activities and competition with a gray wolf and other predators. So please, next. And all these definitions, I believe, they suffer from a very pronounced human-centric approach. So we want to know more about the diseases which can influence our life, our health, directly or through the agriculture or domestic animals, and even antibiotic resistance is viewed from too. And what we will see if we will try to shift this focus and to look what is going on in wild animals from point of view of One Health. What if concept of One Health was formulated by wild animals but not humans? Please, next. And it is very important here also to mention that we have to view this as a ecological process, and in ecological process we view everything in a complex, and ecology of infectious diseases is not a new approach, And if some pathogens are shared between human and domestic animals and exist in a wild animal population, it's clear that you cannot eliminate it and cannot influence on its circulation without understanding how it is transmitted and what are reservoirs and what are the influence of abiotic environmental factors on it. This next one. And altogether, it resembles me classical circus number with the plates, with balance and plates. You have to keep in mind everything, and yeah, it's a good metaphor also for One Health approach. Please, next one. Nothing new in this world, and first. Doctors who dealt with a tropical disease, with a vector-borne disease, they already knew that you have to know the nature reservoirs, transmission routes, and pay attention on the local peculiarities of the disease to understand and to break the life cycle, prevent more people to get ill. And this is an example of Chagas disease or Leishmania, trypanosomiasis, and malaria. Please, next one. And of course, plague. And nothing new in our world. You can see that here pretty well studied, and there is an understanding that there is a circulation in the wild animals, and if we will change the habitats and build our villages or brought our agriculture animals to the area of a natural foci of the disease, the agriculture animals will be influenced, and if you will bring animals to our cities and connect it with a global trade, you will have a pandemic. Nothing new in this world except of a new disease. Please, next slide. What we have learned from last COVID pandemic. Again, there was no such disease a few years ago, but the predecessor of coronavirus existed, COVID-19 existed in a population of bats. And bats probably suffered from it, but not dying out, and there was no major changes in ecosystems due to this disease. At least we didn't document it. Okay, so next, please. Peculiarity of a global ecosystem now is a strong influence of a human on it, and it's not wild ecosystem anymore, not natural ecosystem anymore, but rather ecosystem which was formed under the heavy influence of human activity. And just for example, that if we will count all the animals by heads, all mammals on Earth, 96% of them currently living on our planet will be either humans or our livestock, and only 4% will be wild mammals, and there is no wonder that many of them are endangered and on the brink of extinction, like a rhinoceros depicted here. And it also must influence ecology of pathogens. And even more, if we shift from this approach from mammals to other systematic groups, we will see that the majority of biodiversity comes from invertebrates, from microscopic world. And if we will consider chordates, our relatives, the most of species will live in the water and will be fishes. Next, please. So let's imagine that we are amphibians, and we are intelligent amphibians, and we are trying to have our own One Health approach. So we are endangered. So we are heavily suffering from many things. However, we are important as amphibians. We are important for ecosystem functioning. We are important for science. We produce a lot of interesting things. We are indicators of a healthy environment. We control populations of other animals, insects specifically. We are food to other animals too, and we are important for human culture, but humans are outside of our scope, and medicine. And then some nasty humans start to produce a lot of goods and transfer these goods around the globe, and moreover have a pet trade with amphibians. Next slide, please. And together with the pet trade, we transport many diseases, and one of them, which gain considerable attention because of its impact on the populations of amphibians, is Chytridiomycosis, because of fungus. And starting from approximately 70s, but peaking in late 80s, 90s, it spread across the globe and come to the Central and Southern America and wipe out many species one by one. So this is a terrible episode which causes global changes in ecosystems. And I can bet that most of you never heard about it because you are not zoologists and not interested in frogs, who are not agriculture animals, and only rarely maybe having same disease as the humans or our pets. Okay, next one. So, and surprisingly, there was no data about BD or its relative, another species of a fungus which caused the disease in salamanders. There was no data in Ukraine until last year, and we initiated a project and collected samples across the territory of Ukraine, and now preliminary results showed that territory of Ukraine either free from them, or we are on the process of spreading this disease. It may finally come to our country, and it is spreading slowly just because the density of a populations of amphibians are low, or more dry climate compared to the Central and Southern America prevent fast spreading of a disease. But we have it, and we are preparing publication about it now. Next slide, please. Very similar process had happened with bats. So here you can see two pictures of eastern United States with red circles indicating the size of population of bats in United States before and after pandemic. And before, bats are interesting animals who collect, who gather in a great numbers in the colonies to overwinter, and in this crowded condition, they easily transmit many things from one specimen to another. So prior to 2008, I believe, there was huge numbers of overwintering bats in caves in the eastern United States. And after the mysterious disease called white nose syndrome, bats almost vanished from eastern United States. And this is the situation which was caused by human probably too, because these caves with bats was frequently visited by speleologists, people who research caves, and these speleologists managed to travel from Europe to United States to visit local caves, and they didn't pay any attention to biosafety, and they used the same equipment, the same clothes, the same shoes, went into the cave and caused the disease which take most of the populations there. Please, next. Yeah, it is white nose syndrome because it starts in the winter time, and mold start to grow on the surface of a body of overwintering bats when the immune system is down, and eventually it causes the death of most of the infected animals because of depletion of fat resources. The disease is caused by invasive fungal pathogen Pseudogymnoascus destructans, likely originating from Eurasia. So please, next one. And finally, the same direction has another disease important for reptiles. It's a snake fungal disease, also from probably Europe or Asia to North America. Fungal diseases are usually slow but often deadly. It starts from a skin and may have a systemic consequences, and first it was found in a few states, and now in 2019, according to the data of 2019, it is almost everywhere in United States. Few known about the origin of a snake fungal disease. Most probably it is also from Europe, and only a few recent studies covered European populations of snakes where snake fungal disease is not deadly. Guess why? Because the pathogen and host are often co-evolve, and if they co-evolve for considerable time, the host became resilient to the disease, and disease became less and less severe. Please, next. We also doing a research of snake fungal disease in Ukraine with in a collaboration with Virginia Tech University, and again these are also very fresh results of last 2 years of sampling, that indeed snake fungal disease is present in a few species of native Ukrainian snakes, like Natrix natrix. And now it's very important to understand the ecology of a pathogen, why it is present in certain populations but absent in most of the populations of a species. Is it new transmission? Is it because of abiotic factors? Is it because of a human pollutants in natural water bodies? What is the explanation of observed inequality of species to this disease, susceptibility of a species to this disease? Please, next. And while monitoring local species, we found also some other fungi causing not severe, medium mycosis, surface mycosis in other snakes, and this is Bavaria bassiana. It was reported previously as an occasional pathogen of reptiles. What is interesting with Bavaria bassiana, it is often used by a human as a control agent of insect numbers in agriculture. So somehow also human can influence by spreading, for example, Bavaria bassiana, not only pest insects, but all other animals in an ecosystem if they are susceptible to this disease. Next, please. So what is common between BD, white nose syndrome, and SFD? They are all fungi, and it's also true for a human population that we are expecting some new emerging fungal diseases in years to come. And this is to remind us that we are part of a global ecosystem, and if there is a outbreaks of a mysterious fungi in India, it's not a outlier, and we observe the same in animals last decades too. Next, please. So, and another project we are doing in a collaboration with Ukrainian scientists in Kiev, we do metabarcoding of a snake gut microbiome, which is unique place, interesting place. Snakes are having large intake food, quite rarely every 2 weeks or so. They have a hibernation when they do not eat and stay inactive on a sub-zero or near zero centigrade temperature. They can starve for a long time, and they could serve both as a source of pathogen and reservoir of it. Please, next. So we do first a traditional bacteriological screening, growing bacteria on a media in a petri dish, but it gave us very limited results and take a lot of efforts and resources. Next, please. Next. And metabarcoding approach instead, with a very limited efforts, give us a lot of information about snake microbiome. We extracted bacterial DNA from swabs, from cloaca, and did a standard microbiome protocol. Next, please. So we studied only few snakes, and our results from next-gen sequencing was analyzed on a pipeline, bioinformatic pipeline. Next, please. And what we found instantly that there are a lot of facultative pathogenic genera in a snake gut microbiome, including Salmonella, Enterococcus, Yersinia, Serratia, Obisum bacteria, Hafnia, Klebsiella, Morganella, and they were present in most of the samples. Next, please. And this is a typical representation of a microbiome results. So this pi is a number of reads, and colors and sectors of this circle are number of reads originating from one genus or one species. You can see that there is a majority of reads from one species, and there is ecological rule which describes this diversity. Next, please. There is a strong differences in a number of reads. Probably we have problems with extraction and amplification of 16S of microbial RNA sequence. Next, please. And also number of species revealed from a sample depends on a number of reads from a sample, so methodologically it must be normalized. Next, please. But obviously there is no connection between season and number of bacteria species in a sample. Probably snakes can serve as a reservoir of many of these species we mentioned before. Next, please. Another very strange and surprising result that all samples obtained from different individual snakes appear different. So every snake is different according to its microbiome. Next, please. And only 2 species or 3 genera were present in every sample we studied. So most of the species of bacteria were unique to individual snake. Next. And let's skip it. Yeah. So conclusions from this part of my presentation that the gut microbiome of vipers have many potential pathogenic species, and this diversity does not decrease after a long, almost half-year fasting and hibernation at low temperatures. And microbiome composition has a very high degree of individuality, and we have to continue our research in this direction. Okay, next. So, why we should care about the health of wild animals? Not because we want to everybody be healthy, but we want ecosystem stability. And this ecosystem stability, from one hand, is self-sustained. Ecosystems are reacting and are resilient to some degree. But we are responsible for ecosystem disturbance, and that's why we are observing something which is not natural conditions already now. Next, please. And as a zoologist, I start to promote more safety during our research, and to be more consciousness about the role our study may our research activities may play in natural populations of wild animals, disinfect our tools, change our clothes, and disinfect our tools between individuals, not to cause a spread of a disease, what had happened before and what had reported before with the BD or other pathogens I mentioned today. Next, please. Okay, so that's it. Thank you for your attention. And this is my picture when I was too young to care about the biosecurity. And there was one more slide there with the acknowledgements, but I think, yeah, you can demonstrate it, but we can actually skip it if we don't have time. Thank you. Yep.
We just have to show the acknowledgement because this is very important to acknowledge all members of the team. Thank you, Oleksandr, for your presentation. You brought a lot of interesting food for thoughts for both veterinarians and medical doctors, physicians. And I hope that we can find some projects for collaboration, because there are a lot of issues we can work on together. So once again, thank you.
Thank you for having me.
Thank you so much.